Okay, so I hope you're having your notes. So first of all, what is we're going to discuss about developmental biology, DB. Okay, so first of all, I would like to say what is embryology? Embryology, embryology, embryology. You know what is embryology? Embryology is a study. It is a study of phase. Phase is a time duration. Study of the phase between fertilization and birth. So the changes that is occurring during this time period, that is from fertilization to birth, that can be called as embryology. Embryogenesis occurs during this time period. Embryogenesis, the formation of embryo and the development of embryo, that occurs during this particular time period. That is embryology. And what is developmental biology? DB, developmental biology. Developmental biology. What is developmental biology or DB? You know, that is a study. That is a study of phase study from fertilization to death. So from fertilization to death, that is known as developmental biology. From fertilization to birth, that is known as embryology. In another term, we can tell we are continuously developing. We are continuously developing. We are continuously developing towards what, or up to death. Till our death we will develop. So can you tell any development that is occurring in your body now? So we all are attained our puberty, our growth is completed. Even though our growth is completed, some portion of our body is still growing, right? Which portion of your body is growing now? Mention any part of your body that is still growing.
Hair, nails.
Yes. Yes, I think that was correct. Once again.
Sir, hair, nails.
Hair is continuously growing. Our nail is continuously growing. All these things are growing. Till our death, all these structures will grow, right? So that is developmental biology. And embryology. Now you know what is the difference between developmental biology and embryology. You know, we all were started our life simply with a sperm and ova, right? Sperm and ova. Sperm is a haploid structure, ova is a haploid structure. So the sperm and ova joins together through a process known as fertilization. The sperm and ova joins together and produce a structure. That structure is known as zygote. Zygote is diploid to a number of chromosomes are present there. To the zygote, a process will occur. That process is known as cleavage. When cleavage occurs, you know, cleavage to the zygote occurs. At first, the zygote will produce a structure. That structure is known as blastula. After cleavage, the zygote will produce a structure. That structure is known as blastula. Blastula is a mass of cells. A mass of cells will be produced. That structure is known as blastula. Sperm is a single structure. Ova is a single structure. Sperm and ova joins together to produce a zygote. Zygote will continuously develop and produce a structure. That structure is known as blastula, right? That structure is known as blastula. The process by which the zygote is getting developed into blastula, the process behind that is cleavage, even though that process can be called as blastulation. Through blastulation, blastula will be produced. To the blastula, what will happen? You know, to the blastula, what will happen? A process will occur to the blastula that is known as gastrulation. A process will occur there. That process is known as gastrulation. Through gastrulation, the blastula will be converted into a structure known as gastrula. That is known as gastrula. The blastula will be converted into a structure. That structure is known as gastrula. When we are looking into the blastula, you know, when you are looking into the blastula, Just wait for just wait for 1 minute, okay? Azan is going on. Okay. Mm, So when we are looking into the blastula stage, if you are looking into the blastula stage, uh, the cells will be scattered. Scattered cells will be the scattered cells. Scattered cells will be present in blastula stage, and the cells will be not much organized in blastula stage. Not much organized. Okay, not much organized. Okay. The cells will be scattered in blastula stage. The cells will be simply scattered. So there is a process known as gastrulation. In gastrulation, the gastrula will be produced, right? In gastrula, what will happen, the cells having same fate, 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 what will happen, they comes together, they comes together, they comes together. What does that means, in blastula stage, a number of cells will be there, a number of cells will be there. Which cell is producing eye, that will be here. Which cell is producing our nose, that will be here. Which cell is producing our liver, that will be here. But when it is in coming into the gastrula stage, the cells that is producing our skin, that will come together. That will only come together. The cells that producing a liver cell, that will come together. In that manner, they will form three layers. Three layers will be formed in gastrula stage. Three layers will be there. In gastrula stage, if you are looking, three layers will be there. Which all are those layers, in gastrula stage, if you are looking, you can see an outer layer will be there, an inner layer will be there, and a middle layer will be also there. The outer layer is known as ectoderm. Outer layer is known as ectoderm. The ectoderm layer will be there. The middle layer is known as mesoderm. Middle layer is known as mesoderm. Mesoderm will be there. and an inner layer will be also there. An inner layer that is known as endoderm. So 3 layers will be produced in gastrula stage: ectoderm, endoderm, mesoderm, and endoderm will be produced. From these 3 layers, from these 3 layers only, our organs will be produced. From these 3 layers, our organs will be produced. This organ will mature. This organ will mature. Whenever the organ mature, the organs mature, they again start to produce sperm and ova. So we were started our life from sperm and ova. So this process will continue, and it is known as the cycle of life. This is a cycle of life. Cycle of life. This is known as the cycle of life. It's a cycle, right? This is known as the cycle of life. From ectoderm, if you are looking into the ectoderm, from ectoderm, if you are looking, you can see our epidermal cells. Epidermal cells will be coming from ectoderm. What is epidermal cells? Our skin. Our skin's neurons like things are produced from our ectoderm. Mesoderm will produce our muscle. Mesoderm will produce our muscle. Muscle will be coming from mesoderm. RBC, blood cells will be coming from mesoderm. Such things will be coming from mesoderm. What happens from our endoderm? Endoderm, for example, lung cells are produced from endoderm. Our stomach cells are produced from endoderm. Stomach cells are produced from endoderm. All these things are produced from endoderm. Okay. So once again, if you are looking, sperm were there. Sperm and ova joins together. They produce a zygote, right? Zygote. Then zygote were produced a blastula. In between zygote and blastula, first of all, the zygote will be converted into another structure. The zygote will be converted into a mulberry-like structure. Then only that will produce as blastula. This mulberry-like structure is known as morula. That is known as morula. Morula is otherwise known as little mulberry. Little mulberry. You know mulberry? That is known as little mulberry stage. Morula is known as little mulberry stage. Then from the morula, blastula will be produced. If you are looking into a blastula, when we are looking into a blastula, listen. When we are looking into a blastula, this means when we are looking into a blastula, here it is blastula. When we are looking into the blastula will be arranged in this manner. The blastula will be arranged in this manner. There will be an outer layer of cells and an inner layer of cells will be there. The outer layer is known as trophoblast. Outer layer is known as trophoblast. Whereas inner layer is known as inner cell mass. Inner cell mass. Outer layer is known as trophoblast and inner layer is known as inner cell mass. The inner cell mass will be developing as embryo. The kid will be developing from the inner cell mass. Inner cell mass will be developing as embryo. Whereas the trophoblast, what is the duty of trophoblast? Trophoblast will be attaches on uterus. Trophoblast will be attaching on uterus and inner cell mass will be developing as embryo. So this is the blastula stage. This is a specialty of blastula stage. From the inner cell mass only, you can get these 3 layers. That germ layers. The inner cell mass will be producing the inner cell mass that is only producing the germ layers, ectoderm, endoderm, and mesoderm. The cells in blastula is known as blastomeres. The cells in blastula is known as blastomeres. Blastomeres are the cells present in blastula, and these cells are present as inner cell mass and outer trophoblast layer. Whether it is clear?
Yes, sir.
Clear?
Yes.
I know some students will be from botany, microbiology background. So something will be, some terminology will be new for them. If you're having any doubt, you can ask me, okay? May I move to the next page?
Okay, sir. Yes, sir.
Okay. So we are beginning from sperm and ova. So the first thing, that is gametogenesis. We can discuss from gametogenesis, that is a production of gamete. Gametogenesis, production of gamete, sperm production and ovum production. Sperm production is known as spermatogenesis, whereas ovum production is known as oogenesis. Okay. If you're looking into the cells present in our body, we can classify the cells into two types. There are somatic cells. Cells are of two types. Somatic cells are there. Somatic cells, and the second type of cell is known as germ cells. Somatic cells are there and germ cells are there. These are the two types of cells present in our body. The speciality of somatic cell, in somatic cells, we can see mitosis only. Mitosis. What is the specialty of mitosis? Mitosis means that maintain chromosome number, right? Mitosis will maintain chromosome number. That means if the cell is having 46 chromosome, the two cells that is produced from this single cell will be containing 46 numbers of chromosome. 2n will be the result. This is mitosis. Whereas the specialty of germ cells, the specialty of germ cell is that the germ cells can do another division, that is reduction division. Reduction division. What is reduction division? That is meiosis. There is another division that is known as meiosis. Meiosis is what will happen in reduction division in meiosis, that reduce chromosome number. That means if the cell is having 46 chromosome, the divided cell will be having 23 chromosomes each. That is reduction division, right? That is reduction division. Okay. So the gametogenesis, the gametogenesis means who is doing the gametogenesis? Germ cells. Germ cells only initiate. Germ cells initiate gametogenesis. The process of gametogenesis, that is a duty of germ cells only. Germ cells are doing the gametogenesis. The germ cells are producing. Germ cells will be producing. They will be producing proteins and molecules. They will be producing the proteins and molecules. Sorry, proteins and molecules. Proteins and molecules for gametogenesis. Proteins and molecules for gametogenesis, that is produced by the germ cells. Okay. There is a specialty for germ cell. Germ cells are having a specialty. That specialty is known as autonomous specification. Autonomous specification. Autonomous specification. Anyone of you studying in an autonomous college? Anyone of you studying in an autonomous college?
Yes, sir.
What is the specialty of autonomous college?
So they can frame their own rules and regulations,
Yes,
especially examination pattern and
yes,
all.
yes. In the same manner, the germ cells are having the autonomic specification. What is that autonomic specification? No external signal is needed. No external signal. For a normal college, the university has to decide what is their syllabus. The university has to tell them when to conduct examination. But in the case of autonomous college, they will do as it their own, right? In the same manner, in germ cells, no external signal no external signal is needed. No external signal is needed for their development. For their development, they will develop by their own. For their development, no external signal is needed. Nutrients, proteins, which is required for the development, nutrients, proteins, etc. Nutrients, protein, etc. are already stored in their cytoplasm. If you're looking into their cytoplasm, all the nutrients required for their development, the cytoplasm is known as germ plasm, okay? Germ plasm. As it is a germ cell, the cytoplasm is known as germ plasm. So if you're looking into a cell which is a germline cell, okay, the cell will be containing all the nutrients, all the protein which is required for further development. It is not like ordinary somatic cell. For somatic cell, they want to get protein, nutrient from the external or from which portion that is getting they require that. In cell division, we'll discuss all those things. But here, all the chemical required for their division will be already stored. Some of example for such chemicals are Vasa. Vasa is an example. Vasa. Nanos is an example. Nanos is an example. Tudor is an example. Tudor. PV is an example. PV. What all are these things? They are conserved molecules. They are the conserved molecules. You can expect big part question from this, okay? Conserved molecules in germ plasm. These are the conserved molecules in germ plasm, which helps for autonomic specification. We already told about autonomic specification and gametogenesis. Autonomic specification and gametogenesis. Okay. So this is about the chemical substance that is already stored there. Whether it is clear?
Yes, sir.
Okay. May I wait? May I wait?
Okay, sir.
So, primordial germ cells. Primordial germ cells. It's primordial germ cells, Primordial germ cells, that is PGC. PGC are the progenitors of gamete. The first cell from which the gametes are produced is known as primordial germ cells. Whether it is sperm or ova, they are produced from the primordial germ cells. During our developmental stage, they develop the primordial germ cells, PGC. They develop far away from the gonads. So they will be developing far away from the gonads. During our developmental stage, in our body, the PGC will be produced and the PGC will be developing far away from the gonad. They migrate into the gonad. They migrate into the gonads. And after reaching the gonads, they performs gametogenesis. Then they perform gametogenesis. Okay. So if you are looking into the gametogenesis, from the beginning, you can see 5 things that we are going to discuss about. The first thing is development of the first thing, that is the development of germ plasm and specification of PGC, that we will discuss. Then the second thing, migration of PGC. After that, migration of PGC, that PGC want to migrate. Migration of PGC into gonads. Migration of PGC into gonads. Whether it is testis or ova, they will move. If it is testis, they will become as sperm. If it is ovary, they will develop as ova. Then after reaching into the gonads, they will do meiosis. They will do meiosis. They will do meiosis. After doing meiosis, then there is a differentiation. Meiosis will not only produce the gamete, then there is another process how to occur. We will discuss one by one. Differentiation of sperm and egg. After that, the sperm and egg will be produced by differentiation. Then what we have to discuss the last one, that is hormonal control. Various hormones are behind all this. At hormonal control, we will discuss hormonal control. Then we will discuss about gamete maturation. We will discuss about the maturation of gamete maturation. And we'll discuss about the ovulation, and also spermiation. All these things we'll discuss one by one. Okay. Ready? So, in mammals, if you're looking into mammals, in the case of our mammals, what is the specialty of mammals? Who are mammals? Mammals means what do you mean by a mammal? Hello?
Gives birth to their young ones.
They are having mammary glands, right? Mammals means they are having
Yes, sir.
mammary gland. Arul Jyoti. Hello, Arul Jyoti.
Yes, sir.
Giving birth to the young ones is known as
Mammals.
Who is giving birth to young ones are known as viviparous. Okay.
Oh, okay, sir.
Who is oviparous? Oviparous means laying egg. Oviparous, they lay egg. If they are laying egg, they are known as oviparous. If they are giving birth, that is known as oviparous. And if the young ones are developing, if the egg is developing inside mother's body, that is known as ovoviviparous. For example, in the case of some fishes, fishes, they will lay the egg, but the egg will be present inside mother's body. In the case of some snakes also, they will produce egg, but the egg will be present inside mother's body. The egg will be hatches out inside mother's body. Such things are known as ovoviviparous. Oviparous means which are laying eggs. Viviparous means which are giving birth. Okay, so mammal means they will be having mammary gland. They will be delivering young ones, of course, but not in the case of all mammals. Some mammals are there which are laying eggs. Echidna, platypus, all these are mammals, but they are laying eggs. They will lay egg. The egg will be hatches out outside the body, and after that they will feed the young one. That is the specialty of mammal means they are having mammary gland to feed young one. We all are mammals. Okay. So I have told that there is the progenitor germ cells, PGC will be produced, and the PGC want to be migrated into. PGC will be migrate. The PGC will be migrate into gonads. They will migrate into gonads, right? The gonads can be testis. In the case of males, they will migrate into testis. In the case of female, they will migrate into ovary. In the case of female, they will migrate into ovary. Testis and ovaries. When the PGC migrate towards the gonads, in the case of mammals, the migration with the help of SCF. What is SCF? That is stem cell factor. The migration is done with the help of the factor. That factor is known as stem cell factor. That is a stem cell factor. Okay, clear. I'm wiping this. So we are going to discuss about spermatogenesis first. That is a production of sperm. Spermatogenesis. We can discuss about the spermatogenesis, production of sperm in males, right? That is spermatogenesis. Spermatogenesis occurs in testis, right? That process is occurring inside the testis. We are looking into the testis, the testis is situated outside the body. That is in a particular sac-like structure. That structure is known as scrotum. All these things you know very well, scrotum. Inside the scrotum, there is testis. 2 testes will be there. Not going into the dimensions at all. If you're looking into the testis, the testis will be containing a lot of lobules. That lobules are known as testicular lobules will be there. Testicular lobules will be there. Inside a testicular lobule, if you are looking, the testicular lobule will be containing seminiferous tubule. Seminiferous tubule. There will be seminiferous tubules. Seminiferous tubules will be there. That seminiferous tubule is approximately 800 feet long. Seminiferous tubules are there. Inside the seminiferous tubule, suppose it is a seminiferous tubule. If I am looking into a seminiferous tubule, okay, this is a seminiferous tubule. Its inner side. This is a lumen. This is a middle. This is the lumen. Lumen is the inner portion. Okay, this is that tube. From the periphery, the maturation occurs from the periphery. The sperm maturation occurs from the periphery of the seminiferous tubule. So you can say maturation, sperm maturation. Maturation occurs in the periphery. In the peripheral portion, you can see the maturation. Maturation is occurring in the peripheral portion. And into the lumen, into the middle portion, mature sperms are released here. Mature sperms are released into lumen. Mature sperms are released into lumen. From the periphery, the maturation will occur and the mature sperm will be released into the lumen. Then what will happen, from suppose it is a testis, okay, it is a testis. From the testis, from the tubule, the sperm will be produced, right? The sperm will be flushed outside through these ducts. A number of ducts will be there. Through all these ducts, the sperms will be moved outside. So the ducts include, there will be rete testis. Rete testis will be there. Rete testis. Vasa efferentia. Epididymis. Then vas deferens. All these are the tubes. Through the tubes only, the sperm will move. And at the end, the sperm will go through all these tubes. The sperm will go. So here you can see the urinary bladder. Then urinary bladder. From the urinary bladder, you can see the urethra through which the urine is removing out. Urethra will be here. By covering the urethra, you can see the penis. Penis will be here. This is a penis. So the tube which is ascending, the tube, they will join here as ejaculatory duct. ejaculatory duct. This is known as ejaculatory duct. Anyhow, through all this duct, the sperm will be moved outside. Okay, clear. So we are going to discuss about the spermatogenesis in details. Whether it is clear?
Yes, sir. Yes.
If you're having any doubt, you have to ask me whether my voice is clear. Are you able to?
Yes,
hear me?
sir.
Okay.
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So when a progenitor germ cells, okay, when a progenitor germ cell, when it is reaching inside the testis, okay, when it is reaching inside the testis or ovary, when it is reaching inside the gonads, when it is reaching inside the gonads, now the progenitor germ cell is known as gonocyte. It is known as gonocyte. When the PGC that is reached in gonads, that is known as gonocyte. Okay, that can be called as gonocyte. That can be called as gonocyte. Okay. Ready? So first of all, we can look what will happen to the gonocyte in the sperm production. What will happen to the gonocyte in sperm production? Okay, the gonocyte, that is 2n diploid in number. The gonocyte will multiply. The gonocyte will multiply. They will produce again the gonocyte. They will develop. They will do mitosis and produce a number of cells. This 2n will again multiply. 2n will again multiply. Again they will produce 2n. That is the gonocyte, they will increase their number. They will increase their number. Whenever they are increasing their number, this process can be called as proliferation. This process can be called as proliferation. They are increasing their number, right? This is known as proliferation. And these cells are known as the cells which are doing the proliferation, that can be called as type AS spermatogonium. That can be called as type AS spermatogonium. So a number of type AS spermatogonium is produced now. That is type AS spermatogonium, and the process can be called as proliferation. That is clear? That is proliferation. So these all are AS cells, right? This is AS, this is AS. All these cells are AS cell. So now what will happen to the AS cell? We can check to the AS cell. This AS cell, this AS cell, that will again divide. One AS cell will divide. One AS cell will divide, and that produce another AS and one A1 cell. AS cell will produce type AS spermatogonium, that will produce one AS cell and one A1 cell. A1 cell will be produced. A1 and AS cell will be produced. So in this manner, when a AS cell divides, that will produce one AS, means that will always maintain their population. So this AS cell, what is happening, this cell, what is that? That's a stem cell. Stem cells always maintain their population, always maintain their population. One of the major difference between spermatogenesis and oogenesis. In the case of a female, the ovum production will be stopped, right? Ovum production will be stopped, ceases around the age of 45 to 50 years after her menopause. A female will be reproductively active only between menarche and menopause. Menarche is her first menses. First menses will happen around the age of 10 to 13 years. Menarche. That is known as menarche. First menses. First menses is known as menarche. That is known as menarche. And the last menses is known as menopause. Last menses is known as menopause. A female can produce ova in between menarche and menopause only. Whereas in the case of male, in the case of a male can produce sperm throughout his lifetime. In the case of a female, ovum production stops around the age of 50 years. Whereas in the case of a male, the production of sperm continues even in older ages. The reason for that, the ASL, always when an ASL divide, the ASL will produce one ASL and one A1 cell. This ASL will be stored there. Then from this A1 cell only, he will produce sperm. The ASL, again when the ASL divides, that will produce one more AS and one A1. From that A1 only, he will produce sperm. The ASL will be always conserved. Are you getting my point? Is this clear?
Sir, reply. ഒന്നും കൂടെ ചെയ്യും. Sir, ഒന്നും കൂടെ explain ചെയ്യൂ, sir.
In the case of a female, the major difference between a male and female is that a female will be reproductively active only up to a certain age. Certain age means up to 45 to 50 years of age only, she will be reproductively active. During that time only, she can produce ova. Whereas in the case of a male, the male can produce sperm throughout his lifespan. A male is reproductively active till his death. So a male is reproductively active till his death means he can produce the sperm till his death. Whereas in the case of a female is able to produce ova up to a certain age limit. That means between her menarche to menopause. You may be knowing menarche and menopause, right? Menarche is the first menses and menopause is the last menses. What is happening during menstrual cycle? In a menstrual cycle, what is happening? We will discuss in coming classes. In menstrual cycle, a female is producing ova. In a menstrual cycle, one ova will be released from female's body, right? So the first ova will be produced when female attains her puberty, that is menarche, and the last ova will be released out when the female attains her menopause, that is the last menses. So in between menarche and menopause only a female is active, whereas a male is active throughout his lifespan. The reason for that, a male when he is producing sperm, what he will do, when he is producing sperm from a particular cell, for example, from this cell he is going to produce sperm. This is a AS cell. He will divide that cell in a manner means he will produce one AS cell and one A1 cell. And from this A1 only he will produce sperm. This AS is conserved there. This AS will remain there. Whenever again he is going to produce sperm from this AS cell, he will produce again one AS and one A1. And this A1 will become a sperm, whereas this AS will be stored there. Again, when he is producing sperm from this AS cell, again they will produce one more AS and one A1. This A1 will become a sperm, and this AS is conserved. Again, from this AS, they will produce one more AS and one A1. This A1 will become a sperm, whereas the AS is conserved there. This type of a mechanism is not present in the case of a female, but it is present in the case of a male. Whether it is clear?
Yes, sir.
Okay. So a female will not be reproductively active throughout her lifespan, whereas a male will be reproductively active throughout his lifespan. Okay. So now, what will happen to the A1 cell? What will happen to the A1 cell? That is our question now. What will happen to the A1 cell? This A1 cell, when the A1 cell will divide, the A1 cell divides, the A1 cell divide and the A1 cell will produce another A1. A1 will produce one more A1 and a B cell will be produced. The A1 will produce one more A1 and also a B cell. This B cell, this B cell, till now, what was happening, till now, up to this division from gonocyte to the production of the B cell, all these processes were mitosis. Mitosis only was happening here. From the B cell only, then the B cell only will undergo meiosis. B cell will undergo meiosis. Till now, all the divisions were mitosis. But what happened to the B cell, the B cell will undergo mitosis. B cell will undergo meiosis. So what happened to the B cell, that we can discuss now. What happened to the B cell? So what will happen to the B cell? Listen to me. To the B cell, what will happen? To a B cell, this is a B cell, okay? To a B cell, what will happen to a B cell? The B cell will again undergo mitosis. The B cell will again undergo mitosis, and it will produce a cell that is known as primary spermatocyte. That produces a cell that is known as primary spermatocyte. A cell will be produced there, that is primary spermatocyte. Till B cell, the chromosome number was 46. In primary spermatocyte also, the chromosome number is 46. To the primary spermatocyte, there will occur another division. The division is meiosis. After the meiosis occurs, the primary spermatocyte will produce secondary spermatocyte. It will produce secondary spermatocyte. Secondary spermatocyte will be produced. The secondary spermatocyte is haploid in number, that is containing 23 chromosomes only. That is N, it is haploid. To the secondary spermatocyte, there occurs, this was meiosis 1, first meiosis. Then again, there occurs meiosis 2. Meiosis 2 will occur there. Meiosis 2 will occur there. When meiosis 2 occurs there, it will produce the spermatids. It will produce spermatids. The speciality of spermatids, spermatids is haploid in number. Haploid in number. N number of chromosomes is there. That is 23 number of chromosomes are only present in spermatids. From the spermatids only the sperms are produced. From the spermatids, the spermatozoa will be coming from the spermatids. The spermatids are converted into sperm through the process known as spermiogenesis. Spermiogenesis is a process by which the spermatids are getting converted into spermatozoa. This is a mechanism of spermatogenesis. Whether it is clear?
Yes, sir.
Okay. So when we are looking into the case of a division, in any division that is already mentioned in here, when a 46 chromosome is getting divided into 2 cells, this is not the method of division. When a cell is getting divided, the cell will divide in this manner. In this manner means they will divide into 2, but they maintain, whenever the cells are dividing, they maintain a cytoplasmic ridge. A cytoplasmic ridge will be maintained in between them. So if you are looking into the B cell, when we are looking into the B cell, a number of cells will be there, but the cells will be in this manner. They will be cytoplasmically, they will be connected. All the cells will be connected by a bridge. Why the cells are getting connected by a bridge, Why the cells are getting connected by a bridge? The reason for that, this is a cell, that means this is 44 plus XY, a male, right? 44 plus XY. So this cell will be 22 plus X, and this cell will be 22 plus Y, right? So if they are completely dividing, like this, the protein produced by X chromosome will not be reached here, and the protein produced by Y chromosome will not be reached here. For the healthy development of a sperm, some protein produced from X and some protein produced by Y is required. So in order to make all these sperms healthy, they will maintain the cytoplasmic connection. So what will happen? When they are maintaining the cytoplasmic connection in between them, the protein produced by X, that will reach into this cell. And the protein coming from Y, the protein produced by Y, that will reach here. So both the sperm will develop in healthy manner. So the mainly proteins coded by or produced by, mainly proteins coded by X is needed for spermatogenesis. For sperm production, for sperm production, spermatogenesis, don't think that Y is required. The X chromosome that contains the genetic information or the protein which is required for the production of sperm. So if there is no connection between them, if there isn't any connection between these two, what will happen? This sperm that which contain X will develop into a good sperm, and the sperm which contains a Y will develop into a weak sperm. So when they are maintaining a connection in between them till the last stage, till the final stage, they will maintain this connection. So there will be an equal distribution of proteins between this X and Y cells, so that both the cells will develop in a healthy manner. Whether it is clear?
Yes, sir.
Okay. So anyone give me the answer for why the B cell pool is cytoplasmically connected? Give me the answer, folks. Anyone?
Sir, to produce healthy sperm.
Okay, how is that occurring? How is that occurring? Whenever they are maintaining a cytoplasmic.
Sharing, sir.
Ah.
Sir, by means of sharing the proteins produced by X and Y.
Okay, super. May I wipe now?
Yes, sir.
Okay. So look into a sperm. Okay, this is a sperm. You're looking into a sperm. It will be in this manner. A head will be there, middle piece will be there, and a tail will be there, right? In the head portion, this is a head. Head is there. In the head, if you are looking, you can see a large haploid nucleus. A nucleus will be there. A nucleus will be there. And here a cap-like structure, which is known as acrosome. This is known as acrosome. Acrosome will be the acrosome. Here you can see two centrioles. Two centrioles will be there. One distal centriole. This is distal centriole. Distal centriole will be there. This centriole is known as proximal centriole. Proximal centriole. Proximal centriole is there. This is known as the tail. This is known as the middle piece. Middle piece is there. Middle piece contains mitochondria. Middle piece contains mitochondria. Mitochondria, that is for energy production. Energy production. For energy production, mitochondria is there. Okay. So, whenever the sperm is getting produced, once again, we can look into the things once again. We were having primary spermatocyte. Primary spermatocyte. This is a primary spermatocyte, 2n number. That was divided into produce secondary spermatocyte. Secondary spermatocyte. This is the secondary spermatocyte, that is haploid in number. Then that was produced into spermatids. Spermatids. This is spermatids, that is also haploid in number. Then the spermatids will become as sperm, isn't it? The spermatids will become as sperm. So the process that is occurring here is known as spermiogenesis. Spermiogenesis. That is a spermatid that is looking like a normal cell that is becoming as what? That is becoming as a sperm. So what is happening now? what is happening to the cell? Any message is coming? Are you audible?
Yes, sir. Sir,
Any issue? Kirti?
no, sir.
Okay. So this is a normal cell. This is a spermatid. This is a normal cell. That spermatid is becoming a sperm, right? That spermatid is becoming a sperm. So some events are occurring there. So when a normal cell is getting converted into a structure like this, so what is occurring there? That we can discuss. The first event in spermiogenesis, that is, to the cell, this cell is getting converted into a structure like this, right? So what is occurring there? The first thing is formation of in spermiogenesis. The first thing, formation of acrosomal vesicle. That is the first thing, formation of acrosomal vesicle. So if you are looking into this cell, the Golgi body of the cell, Golgi body will be converted into acrosome. So the Golgi body present inside the cell is converted into acrosome. So that is the first thing. Then what is next thing? The next thing is the condensation of nucleus. Condensation of nucleus. Nucleus want to be condensed. Condensation of nucleus. So for condensation, for nucleus condensation, very well, you might be heard histones are used for the packaging of DNA, right? Histones are used. So here, a histone variant is used to that. That is protamins. Protamins are the histone variant. Protamins are the histone variant used here for condensation. That is, protamins are containing positive charged amino acids will be there in protamins. So sperm nucleus will be condensed. So a spermatid, first of all, in a spermatid, the Golgi body will be converted into acrosome. So in a cell, the Golgi body of that cell will be converted into acrosome. That is the first stage. Then in second stage, what will happen? It is having a large nucleus. That large nucleus will be condensed, right? So that large nucleus in the second stage, this large nucleus will be condensed into a small nucleus. That nucleus will be condensed. For condensation, a packaging is needed. For packaging, protamins are used there. Highly positive histone variant protamins are used there. What is the third step? There is a removal of cytoplasm. The cell is containing large amount of cytoplasm, right? So the cytoplasm will be removed. So the cytoplasm will be removed from this cell, the cytoplasm will be removed outside. Cytoplasm is removed outside. It will be removed in this manner. Small bodies. In this manner, the cytoplasm will be removed. And after that, when we are looking acrosome, a small amount of cytoplasm and the nucleus, it will be produced in this manner. So these small bodies, it is known as residual bodies. It is residual bodies. Cytoplasms are removed in the form of residual bodies. It will be removed as small packets. So, in some cases, the couple, they will be married for more than 2 years. They are having normal sexual practice and all, but they may not be having kids at all. Sometimes the problem will be with these residual bodies. Sometimes, the residual body, when it is removed from the sperm, they will be attached to the sperm. The residual body, when it is attached to the sperm, that will hinder the sperm motility. Such sperms cannot move. If the residual body is attached to the sperm, sperms cannot move. If the sperm is not at all moving, what will happen? They will not be able to move inside female's duct to fertilize the ova. So anyhow, the cytoplasm is removed as residual bodies. Okay, that is the next thing, the removal of cytoplasm. After the removal of cytoplasm, what will happen? After the removal of cytoplasm, Then now it is a cell like this, right? So that is the fourth event. What is the fourth event? You know, that is formation of tail. Formation of tail. Formation of tail. Tail will be formed. Formation of tail from where, you know, from distal centriole. From distal centriole, you know, from distal centriole. From so centriole, you know very well where was the centriole located. So from this, you know, from this, the nucleus is here. Distal centriole is here. From the distal centriole, what will happen? You know, the distal centriole they will produce. Distal centriole will produce microtubule. Distal centriole will produce microtubule. The microtubule will come from the distal centriole. The microtubules which is coming from the distal centriole, that will form the tail of the sperm. That will produce the tail of the sperm. Whether it is clear?
Yes, sir.
Can anyone explain the events in spermiogenesis? Have to call anyone? Okay, Amina.
Yes, sir. First, formation of acrosomal vesicle. அதில் வந்து கோல்கி பாடி தான் ஆக்ரோசோமா ஆக்சுவலாக கன்வெர்ட் ஆகும். நெக்ஸ்ட் வந்து என்னன்னா கண்டென்சேஷன் ஆஃப் நியூக்ளியஸ். நியூக்ளியஸ் வந்து ஆக்சுவலாக வந்து அதில் வந்து புரோட்டாமைன்ஸ் அப்படிங்கிற ஹிஸ்டோன் தான் வந்து நியூக்ளியஸ் வந்து கண்டென்சேஷன் ஆகிறதுக்கு ஹெல்ப்ஃபுல்லாக இருக்குது. அதுக்கப்புறம் சைட்டோபிளாசம் வந்து நிறையா இருக்கிறதுனால அது ரெசிடியல் பாடிஸாக ரிமூவ் ஆகுது. அதுக்கப்புறம் இப்போ ஃபார்மேஷன் ஆஃப் டெய்ல் எப்படி ஃபார்ம் ஆகும் அப்படின்னா டிஸ்டல் சென்ட்ரியோல்ஸ்ல இருந்து டிஸ்டல் சென்ட்ரியோல்ஸ் வந்து மைக்ரோடியூபுலா கன்வெர்ட் ஆகி மைக்ரோடியூபுல் வந்து டெய்லா கன்வெர்ட் ஆகுது சார்.
In any ways that residual bodies are related to infertility?
எஸ் சார். இஃப் த ரெசிடியல் பாடிஸ் வந்து ஸ்பெர்மோட போய் அட்டாச் ஆச்சு அப்படின்னா அது மொட்டிலிட்டியை வந்து அஃபெக்ட் பண்ணும்.
Okay. So there were a proximal centriole, right? There were a proximal centriole. What will happen to the proximal centriole, you know? The proximal centriole, what will happen to the proximal centriole, you know? That proximal centriole will be transferred to egg. During fertilization, the proximal centriole will move into the egg. Along with the nucleus, the proximal centriole will also move into the egg. What will happen to the distal centriole? We have already told about the distal centriole. In detail about the distal centriole. If you are looking at the distal centriole, you know, distal centriole will act as microtubule organizing center. That will act as microtubule organizing center. Microtubule organizing center. That means microtubule will be coming from the distal centriole. It will be acting as a microtubule organizing center. That means they polymerize microtubule. They polymerize microtubule and form the tail. They will polymerize the microtubule and they will form the tail. Okay. The microtubule, if you are looking into a microtubule, you know, if you are looking into a microtubule, the microtubule contains alpha tubules and the microtubules contain alpha tubules and beta tubules are present in a microtubule. If you are looking into a sperm tail, okay, in a sperm tail, if you are looking into a sperm tail, suppose it is a cross section of a sperm tail, okay, it is a cross section of a sperm tail. How the microtubules are organized here, you know, the microtubules are arranged 9+2 arrangement. 9+2 arrangement. What is 9+2 arrangement? You know, the 9 will be present as doublet. 9 doublets will be present there. 9 doublets and 2 singlets. 2 singlets will be present as microtubules. Listen here. 2 singlets will be present. So at the center, you know, if you are looking, you can see 2 microtubules at the center. This is a sperm tail, okay, cross section of a sperm tail. At the center, you can see two singlets will be present. In the periphery, if you are looking, you know, in the periphery, you can see doublets will be present there. Doublets will be 9 in number. 9 doublets will be present there in the periphery. Here it is 9 doublets, and these doublets will be present in this region, and 2 singlets will be present in the middle. In middle, you can see 2 singlets, and center you can see in center you can see 2 singlets, and outer side you can see doublets. They are connected with the help of, this will be connected with the help of, they will be connected with the help of nexins. Will be connected with the help of nexins. Nexins are used for, that is a motor protein. Dynein motor protein. That is negatively directed. They are negatively directed. And they are having, they have ATPase activity. They have ATPase activity. The ATP will be coming from where? ATP will be produced by the mitochondria, and that ATP can be digested by this tail. So whenever they are breaking down that ATP, energy can be produced. So the doublets are connected with the help of nexin, and nexin contains a motor protein that is known as dynein motor protein. The dynein motor protein is having an ability, that is ATPase activity. So 2 type of motor protein, dynein and kinesin. Dynein and kinesins are 2 type of motor proteins. 2 type of motor proteins present in our body, that is dynein and kinesin. So the nexin contain dynein. Nexin will be, the nexin is used for connecting the doublet, and the nexin contain a motor protein that is known as dynein. It is having ATPase activity. ATPase activity means that can digest ATP. ATP will be broken down into ADP and phosphate. Whenever they are broken down into ADP and phosphate, they will release what? Energy, right? ATP is the energy currency of the body. So they will be releasing energy. That energy can be used for sperm movement. But is clear? Clear?
Yes, sir.
Okay. Suppose if dynein is mutated, if in a person dynein is mutated, what will happen? If in a person dynein is mutated, what will happen? In a male, if dynein is mutated, what will happen? Come on, anyone.
Sir, 9 doublets won't be connected, sir.
So what will happen? What will be the ultimate result? That person will be? That person will be? Come on.
Unfertile.
That person will be infertile. Not unfertile, infertile. Okay. So if dynein is mutated, it will result in a disease that is known as what is the specialty of the disease? Sperms are, that person will produce sperm, but the sperms are immortal. The sperm will not move. The sperms are immortal because the dynein cannot hydrolyze. Dynein is hydrolyzing ATP. So in that person's body, dynein will not hydrolyze. As a result, the sperms will not move in that person. So he will be infertile. Okay. So dynein that is needed for, dynein is needed for flagellar and cilial movement. flagellar and cilial movement is done with the help of dynein. Dynein is a motor protein that has ATPase activity that is required for flagellar movement and also for cilial movement. Flagellar movement we can see in the case of sperm. Sperm is moving with the help of flagella. Where you can see ciliary movement in our body, ciliary movement. Cilia is present in our lung, right? In our lung, you can see cilia. So the dynein mutated, it will produce, it can cause bronchial infection. In our respiratory tract, it may cause bronchial infection. Bronchial infection also the result. So in our respiratory tract, the waste materials are removed with the help of the cilia. Cilial movement only helping for removing the foreign particles from our respiratory system. So if the cilia is not working, if dynein is mutated in a person, his cilia will be also not working. Another disease that is situs inversus disease. What is situs inversus? Situs inversus is a disease that is in such persons, the heart in a right side of the body. Normally, our heart is present in the left side of the body, right? So in situs inversus, that is situs inversus, that is helping. Who is helping for the heart cells are moving. Heart cells are moving to left. In our body, the heart cells are present in left side, right? Heart cells are moving left with the help of nodal cilia. With the help of nodal cilia. So if mutation is occurring, the heart will not move into the left, and it will cause a disease. That disease is known as situs inversus disease. That is known as situs inversus. Whether it is clear? Okay. So
Sir, first write பண்ண disease name மட்டும்.
Cartagena triad syndrome. Cartagena triad syndrome. Okay.
Sir, once again.
Cartagena triad syndrome or Cartagena triad disease. Okay. So the days required for spermatogenesis. The days required for spermatogenesis for the cell to become as a sperm, for a single cell to become as a sperm. Any guess? Give me any guess. What will be the time required for the development of a single cell into a sperm? Any guess? One second. Who is this? Shah Sociology? Who is this? Everyone, please unmute yourselves. Kirti, Amina?
Yes, sir.
Harini, Sudipti,
Yes, sir.
Subha.
சொல்லுங்க sir.
Then who is this one? Sudipti Shah. Who is this Shah? Subhalakshmi Shalini. Who is this Shah? Hello?
Sir, don't know.
Sir.
Hello, Shah. Please unmute yourselves. Everyone, please unmute.
Sir.
Everyone,
Sir.
don't mute yourself. Keep in unmuted condition.
Okay, sir.
Yes, sir.
So only this is Shah. This person is not unmuting. Who is this Shah? That person is not in the class also. Hello? Any one of your companion? Do you know?
No, sir.
No, sir.
Okay. Okay, let me remove that person then. Once again, I am asking whether anyone is using others' mobile. Shah? Hello, Shah? Okay. Even though that student is belonging to our class, he or she is not responding, not unmuting. Okay. Hey, how to remove? Okay, removed. Okay, others, please guess what will be the time required for the development of a single cell into sperm? Any guess you can tell. Amina,
Don't know, sir.
Arul,
No idea, sir.
any wild guess is okay because we have already told about all the divisions, right? What is happening? First of all, ASL will be there. Then the ASL will become as B cell. B cell will become as A1. Sorry, AS will become as A1. A1 will become as B. B will become as primary. Primary will become as secondary. Secondary will become as somatid. Somatid will become as in that manner we have discussed. So just tell any guess. Any guess is okay. Oh, that was Shajida. Okay, please rejoin then Shajida. Okay, any guess, any guess, okay. Anyhow, if there is no guess, I will tell. That requires for the development, you won't expect 64.5 to 65 days. That is required for the development of a sperm. 65 days. Okay. 65 days are required for the development of sperm. Ready. So that girl couldn't join again. Sorry. She was not responded then. Elaborate, whether you are having a problem to join in the class today? Elaborate is not here now. She also left put a message like she can't join. What happened? Oh, heavy rain. Okay, ready. Okay, then when for this question you have to answer me. A sperm. This is a sperm. This is a ova. Sperm and ova join together, and they produce a zygote. They produce a zygote. What is the normal fate of the zygote to develop into male or to develop into female? Tell me. Sperm and ova join together. Sperm and ova is fused together. Then they produce a zygote. They produce a zygote also. So I'm asking what will happen to the zygote, whether it will develop into a male or whether it will develop into a female. Come on. Hello? Hello, you're here?
Yes, sir.
Okay. Sperm and ova joined together. That was produced into became developed into a zygote. So I'm asking what will happen to this zygote, whether it will develop into a male or whether it will develop into a female. How that is determined?
Depends on the X chromosome present in the sperm.
Okay, who is replying to me?
Amina, sir.
Okay, Amina. So the sperm contains the sperm is 22 plus Y and the ova is 22 plus X. So what is the normal fate of the egg? What is the normal fate of the egg? Amina.
Male.
Any other answer? Kirti, what do you think? Kirti,
Sir.
what will happen to the zygote?
Male, sir.
Male. So you won't expect the fate of every zygote is to develop into female. Each and every zygote will develop into female only. Okay. Irrespective of it is containing X or Y, the zygote will start its development as female. So even me, during my initial stage, I was a female. So let me explain about this. A zygote is having its normal fate towards the development of a female kid only. Okay, listen how I am explaining the thing. In Y chromosome, Y chromosome, this is a Y chromosome. The Y chromosome contains one region known as SRY region. That is a sex determining region, SRY. That produce one antigen. From SRY, one antigen will be coming out. From SRY, one antigen will be coming out. That antigen is known as HY antigen. The name of that antigen is HY. That is coming from SRY. The HY will be present. Where the HY will be present, I hope you remember about the PGC, progenitor germ cell. The PGC, over the PGC, the progenitor germ cell over the PGC, the HY will be present. The HY antigen will be present over the PGC. If it is containing Y chromosome, the HY antigen will be present outside. So what will happen, if a PGC contains HY antigen, what will happen, the PGC, when it is reaching into the gonad, the PGC, when it is reaching into the gonad contain, irrespective of whether it is testis or ovary. Gonad means it can be testis or it can be ovary. When the gonad is reaching, when the PGC is reaching the gonad, the gonad contains a protein. That protein is known as dazzle protein. Whenever the PGC is reaching the gonad, the HY interact with the dazzle protein. When the HY interact with if the HY interacts with the dazzle, if the HY interact with the dazzle, what will happen, if the HY interact with the dazzle, what will happen, the gonad will develop into what, the gonad will become as testis. The gonad will become as testis when, in the 7th week. In the 7th week of development. The 7th week of development, the gonad will become as testis. Otherwise, what will happen, the default fate, otherwise, otherwise the gonad will become as what, the gonad will become as ovary. The gonad will become as ovary in the 9th week. In the 9th week. So, after the sperm and ova fuses together, we got the zygote. So, whenever the sperm and ova fuses together, we got the zygote. Even though we got a zygote, while simply looking into the zygote, we cannot determine whether it is going into develop into male baby or female baby. When we can determine or when we can recognize that, that is only in the 7th week of development. So, in first week, second week, third week, fourth week, that means 1 month. 5th week, 6th week, 7th week. So after 1.5 months of the embryonic stage only, what will be happening, the PGC, that is a progenitor germ cell, it is reaching into the gonad, and the gonad contain dazzle protein. If that PGC is containing Y chromosome, that will be having HY antigen on its surrounding, on its surface. If that HY antigen is interacting with the dazzle protein, then only that PGC will develop into testis. Then only that PGC will convert the gonads into what? The gonads into testis. Otherwise, the normal fate of that gonad is to develop into ovary. So in my body, in my father's body, in your father's body, in everyone's body, our original fate was developing into a female. All of us, we all are having a female inside our body. A female thing is hidden in our body. So every structure that is, so normally we have developed all these structures in according to female's body only. Then what will happen, for example, in a female, there is the oviduct. The oviduct is coming from the Mullerian duct. The Mullerian duct is developing into the Mullerian duct of a female that will develop into oviduct. So what will happen in the case of the Mullerian duct will be present in every embryo. Every embryo will be contained in the Mullerian duct, which will be developing to oviduct. So what will happen in the case of a male, a male will produce a particular hormone that is known as AMH, anti-Mullerian hormone. Whenever the anti-Mullerian hormone come, the Mullerian duct will not develop into oviduct. That will be developed into what? That will be developed into structures like, in earlier I have told about retia testis, vasa efferentia, seminiferous tubule, in that manner. So every structure in the case of a female is equivalent to a structure in the case of a male. For example, if you are looking into the what I can tell, clitoris of a female. Clitoris of a female is actually, that is in a female, the clitoris is developed, right? That clitoris is becoming, whenever there is a Y chromosome and all these things happen, that clitoris will enlarge and that only produce penis in a male. So penis in a male is equal to the clitoris in a female. The end portion of clitoris is known as glans clitoris. The end portion of penis is known as glans penis. They are homologous structures. The mons pubis in the female, the mons pubis, which is required to regulate temperature, that is a mons pubis, that region is equivalent to scrotum region of a male, that is regulating temperature. In that manner, if you are looking, each and every structure in a female that is present in male. Whenever the development of that structure is hindered, whenever the development of that structure is restricted due to the presence of this anti-Mullerian hormone, then only the development of male structure occurs. Whether it is clear? Amina?
Yes, sir.
Clara?
Yes, sir.
So we all were?
Yes, sir.
So we all were?
Female.
Female.
We all were females. Whether it is a new thing to you?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. So இப்போ போய் வீட்டில் எல்லாருக்கிட்டே சொல்லி கொடுங்க. We all were females. So all these structures present there is present in a male's body, but in an alternative manner only, in a different structural format only. Everything that is present there is present in a, so normally from which region the testis is getting developed. So normally from which region the ovary is getting developed, that region is getting converted into testis. Normally from which region Mons pubis is getting developed, that region is getting converted into scrotum. Normally from which region the clitoris is getting produced, that region is developed into penis. Normally from which region the glans clitoris was produced, that region was converted into glans penis. Clear?
Yes, sir.
Okay. So for female fate, we have to wait up to the 9th week. The female fate will be produced only after if there is in the interaction occurs. HY, dorsal, if all these interactions are not occurring, then for in the 9th week, normally that zygote will develop into a female. Otherwise, the female growth is checked or the female growth is prevented at the 7th week of pregnancy, and that structure will develop as a male. Shajida, whether it is clear for you, Shajida? Shajida will be typing in the text box, right? Okay. So now, for a spermatogonia, to develop into a sperm or spermatocyte, for a spermatogonia to develop into a spermatocyte, that is influenced by two factors. Two factors are behind this. The first factor is GDNF. That is the first factor. That is glial cell line derived neurotrophic factor. That is the first one. Glial cell line derived neurotrophic factor. That is the first one. The second one is SCF. That is stem cell factor. SCF, that is stem cell factor. So stem cell factor and glial cell line derived neurotrophic factors, they are required for the development of spermatogonia into spermatocyte. Higher amount of GDNF, always maintain, higher amount of GDNF always maintains spermatogonia, higher amount of GDNF always maintains spermatogonia. That means stem cell pool. If there is higher amount of GDNF, what will happen? The sperms will not be produced. The cells will produce more amount of spermatogonia, from which the sperm can be produced during favorable condition. So decreased amount of GDNF. What will be? That will be helps in the transition. Helps in the transition means the spermatogonia will be converted into sperm when there is very lesser amount of GDNF. Whenever there is a higher amount of GDNF, the cell will continuously proliferate to produce more amount of spermatogonia. I hope that is clear. Now, inside the seminiferous tubule, I hope you are remembering the seminiferous tubule. Inside a seminiferous tubule, if you are looking inside a seminiferous tubule, you can see two type of cell. The first type of cell is known as Sertoli cell. Sertoli cell. What is the duty of Sertoli cell? Sertoli cell, nourishment to sperm. Nourishment to sperm. That is the duty of the Sertoli cell. And there is male germ cells. Male germ cells. Male germ cells. Male germ cells, or otherwise that is known as spermatogonia. Male germ cells or spermatogonia. The duty of spermatogonia is to produce. They produce sperm. They produce sperm. So outside the seminiferous tubule, outside the seminiferous tubule, you can see another type of cell that is known as Leydig cell. Leydig cell is present there. The Leydig cell produce hormone. Leydig cells produce testosterone. They produce testosterone. The Leydig cell is otherwise also known as interstitial cell. Interstitial cells. That is another name of the Leydig cells. Interstitial cell. They produce testosterone. That is the duty of the testosterone. Okay. So we have to discuss about the hormonal control also. So with that, we can wind up. The Sertoli cell is having another duty, Sertoli cell is phagocytic. Sertoli cell is phagocytic. Phagocytic. That is, they engulf defective sperms. They engulf defective sperms. They engulf defective sperms and residual bodies. What were residual bodies?
Cytoplasm.
What is that?
Sir, division the cytoplasm.
Okay. So look, now the last portion of today's discussion, that is hormonal control. We can discuss about hormonal control also. One thing, I'm sorry that in this week we'll be winding up today because I'm not physically okay. So I couldn't get 2 classes in this week. So Tuesday was my day, but I couldn't take because I was ill. So I hope next week, if I'm taking rest for the coming days, I hope it will be okay in coming days. Okay, so bear with us. For this week, we'll be winding up with these 3 classes. Okay. I hope you are applying for CSIR. Do that immediately. If you are applying for immediately, then only you will be getting the first option as your exam center. When you are lagging, you may be pulled to the second or third center. So do that immediately and make sure that you are having the community certificate with you. If not, apply for that and get it soon. Otherwise, after we are clearing, it will make a big problem. So make all the certificate available and apply. If you are having doubts and all, you can ask. Okay. So look for the hormonal control also from our hypothalamus. A hormone will be coming. That hormone is known as GnRH, gonadotrophin releasing hormone. When the GnRH hormone is going into the anterior pituitary, when it is going into the anterior pituitary gland, the anterior pituitary will produce 2 hormones. The anterior pituitary will produce 2 hormones. The first hormone is known as FSH, follicle stimulating hormone, and the second one is known as LH, luteinizing hormone, which is also known as interstitial cell stimulating hormone, ICSH, interstitial cell stimulating hormone or luteinizing hormone or FSH. I hope you know the full forms and all. FSH is follicle stimulating hormone. Follicle stimulating hormone is FSH. GnRH is gonadotrophin or releasing hormone. Gonadotrophin releasing hormone. Or is LH is luteinizing hormone. That is LH, which is also known as ICSH, interstitial cell stimulating hormone. Okay, leave that. So FSH will act on Sertoli. FSH will act on Sertoli cell. FSH will act on Sertoli cell. This LH will act on Leydig cell. LH will act on Leydig cell. LH will act on Leydig cell. The Leydig cell will do the production of testosterone. From Leydig cell, testosterone will come. Testosterone will come. And the Sertoli cell that produce certain factors. Sertoli cell will produce certain factors. The testosterone will do the spermatogenesis. Testosterone will do the spermatogenesis. Sperm production is the duty of the Sertoli testosterone, whereas the duty of the factors, that is spermiogenesis. Spermiogenesis. What was spermiogenesis? Spermiogenesis. What was spermiogenesis? Tell me. What was spermiogenesis?
Spermatids is converted into.
Spermatids is converted into sperm. That was spermiogenesis. So testosterone is doing the spermatogenesis, whereas spermiogenesis is the duty of the factors which is coming from the Sertoli. Sertoli will also produce from the Sertoli. Sertoli will also produce inhibin. Sertoli will produce inhibin. What is the duty of inhibin? Inhibin will inhibit the andropituitary. That is a negative feedback. Negative feedback. They will do the negative feedback. What is a negative feedback? This is already in progress, right? The reaction is already in progress. The process is already in progress. The Sertoli got FSH. Whenever the Sertoli got FSH, Sertoli will send a message to the andropituitary, that is inhibin, and tell the andropituitary that now don't produce FSH again. We are already working with the FSH that you have already provided to us. That FSH is required now for the production of the sperm. Then after the complete cycle of the production, then only we require the further amount of FSH. So for that particular time period, just stop the production of FSH and LH. That will be going from the Sertoli cell. The Sertoli cell can inhibit the anterior pituitary. So to control this negative feedback is to control the hormone production. To control the hormone production. So what is the difference between negative feedback and positive feedback? Positive feedback in the sense, whenever the message is going, the hormone production is stopped, right? If the hormone production is continuing, that is positive feedback. Whenever a message is going, and if the production is again increasing, that is positive feedback. Whenever the message is going, if the production is decreased, that is known as negative feedback. For positive feedback, if I am telling an example, how long a mother can feed a baby? How long a mother can give milk to young man? How long? The years. Amina.
Don't know, sir.
But what do you think? How long in years? Yes. Up to?
2 years, sir.
Up to 2 years. As according to our religion will think it is 2 years, right? Any other answer? What according to Arul Jyoti? How long? Any mothers are here in the group? Any mothers? No mothers. Arul?
Sir, up to 1 year.
Up to 1 year. What will happen after the next day? After the last day, what will happen?
Sir, food அந்த மாதிரி கொடுத்துக்கலாம்.
Yeah, we can give the food and all. I'm just asking about the thing. Even though we are giving food to the baby, even though we are giving food to the baby, milk will be there, right? I'm asking about the thing. How long the milk will be there in the mammary gland? I'm asking that question. Amina told about 2. So if I'm asking to Amina, what will happen after the last day of second year? After that 365th day, what will happen?
Inhibition.
Amina?
Kerala, sir.
No. Kirti, what do you think?
Sir,
No,
maybe
sir.
negative feedback, sir.
Negative feedback means negative feedback. Okay, in the sense. So there is a positive feedback there. In mammary gland, that is positive feedback. There is no negative feedback. There is positive feedback. Positive feedback means whenever the child is sucking on the nipple, whenever the child is sucking the nipple, the message will be sent to the hypothalamus to release more amount of prolactin. Prolactin is milk producing hormone. So whenever the baby is drinking or whenever the baby is trying to suck the nipple, it will send a message. Why I am explaining this is only to tell about the positive feedback mechanism. In this case, you can see negative feedback mechanism. Positive feedback means whenever the child is continuously trying to get the milk, it will be sending message to the central nervous system to produce more amount of milk producing hormone. So it can be extendable up to 1, 2, 3, 4, 5, 6, 7. It can be continued till they desire to stop the feeding. Whether it is clear? That hormone is prolactin. Prolactin will be produced when the baby is trying to feed. That is positive feedback mechanism. Whenever the process is happening, more amount of hormone is coming. That is positive feedback. Whenever a message is going, if the production is decreased, that is negative feedback. Whether it is clear?
Yes, sir.
Amina, so how long baby can be given the breast milk? Amina? Why I'm
No,
asking to Amina? Because Amina is showing the first name is Amina's name. That's only okay. So that is up to our wish. That is up to our wish only. Okay, it is clear. It is not 1, 2, 3, or 4 or 5 years. Whenever they want to stop, we have to stop. We can stop. But whenever the baby is trying to suck the milk, it will be again sending message for the production of milk producing hormone prolactin, and it will be again synthesized. But it is clear.
Sir, which means prolactin is produced throughout the life, sir.
No. Whenever the baby is, whenever we are thinking that, okay, it's enough, okay, then the baby is not sucking the nipple, the prolactin will not be produced.
Okay, sir.
So that is only happening after 2nd year. So normally in our cases, after 1 or 2 years, what we will do, we will be doing more solid food to the baby, and we will abstain the baby from sucking the nipple, right? So at that time, whenever the baby is not sucking the nipple, the prolactin hormone will not be produced. So if there is a stimulus, then the hormone is coming. That is positive feedback. If there is a stimuli, if the hormone is not coming, that is negative feedback. In this case, the stimuli is inhibin. When the inhibin is going, the hormone production is decreasing. That is negative feedback. Was this clear?
Yes, sir.
So in today's class, we have discussed about spermatogenesis in detail. It's only about the spermatogenesis. We have discussed all the topics, all the things from Gilbert that we have to discuss from spermatogenesis. I hope the class was useful for you. So let me stop the recording first.